Hi, everyone. How are you doing today? Thank you all so much for joining. We are just going to give it a minute. I know that some of you were all in the waiting room, so just give a minute for everyone to move over to the webinar platform itself. But thank you so much for joining us today. We really appreciate it, and we are looking forward to today's discussion. Um, So today, as you guys have seen, probably as it was advertised, our webinar, we're going to kind of focus on the variant classification and reclassification process. Kind of give a brief overview, and then we'll talk a little bit more about what pieces of information may go into classifying the variant. So as a reminder, GenomeConnect is a patient registry, but we do have a number of educational resources available. And so today's lecture is going to be kind of also, our webinar is going to be building on some of our past webinars that we have hosted. So this year in July, we did a whole overview of GenomeConnect. So if you're interested in learning more, check out the video for that webinar. In April, we did a webinar on how to read a report, which really goes through different key pieces of information to review on the report. And then again, we have a number of educational videos that go through an overview of genetics, overview of different types of variants, all really, I find them can be really helpful in taking some of these more complex genetic topics and breaking them down to help everyone understand a little bit more about the genetic information that you were seeing and reviewing. So our YouTube playlist is GenomeConnect and patient data sharing, and the link was for the playlist has been put in the chat. So thank you to all of those who have registered and provided questions. We will try our best to kind of connect and go over some of those questions during the presentation. If you do have questions that come up, feel free to put them in the Q&A box, and that should be towards the bottom right-hand side of the Zoom. If you have any questions, we have a bunch of people here with us to also help out if there are any issues. Also, as a reminder, I forgot to say, but on the slide, all of our webinars are recorded. As you can see, they are then shared with our YouTube channel. And because they are shared on our YouTube channel, because they are recorded, we do recommend not putting your personal information in the Q&A box to try to keep it more to general questions. I don't want anyone to share their own protected health information on a recorded webinar. So this is today's team. Let me start with introducing myself. My name is Caitlin, and I'm one of the genetic counselors involved in GenomeConnect, and we are joined by two other team members. We have Jules, who's another genetic counselor on the team, and Lizzie, who's our GCA on our team, and they will both be here helping us with any, hopefully none, but any IT issues that inevitably come up. And they will also be helping to answer some of the Q&A questions, and then also letting me know if there's any questions that you guys come up or anything that we need to go over. So again, feel free if you have any questions, just put them in that Q&A chat as we go along. So what are we going to do today? So today we are going to kind of provide a brief overview of the variant classification and reclassification process. As I said, this webinar is going to be recorded, and this webinar is really created for individuals that have had genetic testing. So if you do have specific questions regarding your own test results, we encourage you to contact your healthcare provider or a local genetics provider. And we ask to kind of that you don't put your private genetic information in the chat. So I'm going to start with a quick review of genes and variants. And for more information, we do have all of those educational videos on our playlist. As I mentioned, just in April, we did kind of this big review of how to read a report that goes through the types of variants in more detail. So each one of us has more than 1 trillion cells that make up our bodies. We have different types of cells, including our skin cells, our brain cells, and heart cells that are the building blocks of our body. So in each of our cells, all of our genetic material or DNA can be found. Our DNA is like our body's instructional manual. Each instruction is called a gene, and each gene codes for a protein. To keep our DNA organized in our cells, it is wrapped tightly into structures called chromosomes. When an individual has genetic testing, the laboratory is searching for changes, or another way to say changes, genetic variants in their DNA. While most of our DNA sequences are the same from person to person, we do all have variants. Some variants have the potential to impact our health, and these are what we would refer to as maybe pathogenic variants. And other variants do not, and these are what we would refer to as benign variants. Variants classified as uncertain, or sometimes referred to as unclear or unknown significance, often abbreviated as a VUS, do not yet have enough information available to determine whether they are disease-causing, which again is pathogenic, or just a benign genetic variant that each of us have. When a genetic testing laboratory identifies a genetic variant through testing, the next step is to classify that variant. To classify a variant, first we need to understand the gene in which the variant was identified. Then we need to understand the impact of that variant has on the protein. Does it impact the protein's function? Does that then lead to certain health conditions? So there's a number of kind of steps that are dependent on each other before we do get to the specific classification of that variant. Unfortunately, there are many uncertain results, and we do have much to learn in the realm of genetics and health. We know that the genetic basis of between 6,000 to 13,000 genetic conditions remains to be identified. We don't know the genetic implications or cause yet. And then between 16 and 37% of patients who undergo genetic testing in the past few years have received an uncertain result, that VUS, that uncertain, unclear variant of significance. So really, we have much to learn to better answer these questions. And the first thing we need to start with is understanding the genes and understanding how those genes may play a role in our health. And then we can go down to the specific change or variant in that gene. So, as I just said, first question is there enough evidence that variants in this gene can cause this disease? And so in order to do that, we need to understand and study the gene. So a curator or a researcher will gather evidence from published studies about the gene, including genetic evidence from individuals who have been identified to have variants in this particular gene, try to understand, are there any effects on their health? The researcher, the curator, will also look to see has there been any experimental studies done or published regarding this gene's function. The curator researcher will then score the strength of this genetic and experimental evidence for or against a relationship with the disease. So based on the available evidence, a classification is then assigned to describe the strength of the evidence for that gene-disease relationship. If a gene's role in disease has been demonstrated in the research and clinical setting and upheld over time, so over a number of years, that gene will likely reach what we would call definitive or strong, meaning has a definitive or strong relationship to a certain health condition. From there, there are lesser evidence. From there, as lesser evidence is available, then that kind of classification may be different. So we may have limited or no known disease relationship because the score of the evidence does not reach kind of the more definitive state. These classifications of this gene-disease relationship can help guide genetic testing and variant classification. Variants without well understood gene-disease relationships, such as if a variant is found in a limited, disputed, or refuted classification, all those variants may be classified as a VUS, as unknown, because we don't know what the gene does yet. So then we can't even determine the impact of that variant, that protein, on health if we don't know what the gene is doing. Variants with a classification with a gene, variants in genes with a moderate gene disease relationship or higher. So if the gene disease relationship is definitive, strong, or moderate, then the laboratory can really start to tweeze out the specific change, specific variant, and understand a little bit more of how that may impact someone's health. So laboratories may use this type of information, this gene disease validity classifications, to decide what genes they may put on a testing panel, or what genes they may want to report out findings for if someone's doing genome or exome testing. And clinicians may use this type of information when interpreting test results from their patients. Help provide some guidance when answering these questions. The Clinical Genome Research Project, or ClinGen, was created. So ClinGen, as we have kind of talked about a little bit in the past, is an NIH-funded project. GenomeConnect, the patient registry that we work on, is ClinGen's patient registry. We are a very small part of ClinGen. But as I said, ClinGen is dedicated to kind of building this authoritative resource that defines the clinical relevance of genes and variants for use in precision medicine and research, and ultimately hopes to understand what genes and variants are associated with disease and how genetic information can inform clinical care. So as part of this, ClinGen has created a number of expert panels that systematically review genes and publish that gene disease validity score, that classification, that definitive, strong, moderate, limited, so that classification can be used by other researchers, laboratories, and clinicians. These expert panels are typically what I'm going to refer to throughout the rest of the talk are GCEPs, which stands for Gene Curation Expert Panels. We have GCEPs that are curating genes across many disease spaces. This slide that you're seeing is not an inclusive list. This just shows some of the current GCEPs that are actively reviewing genes to date. So next up is variant classification. If the gene has been associated with a health condition, it has that gene disease relationship of moderate, strong, definitive, then the specific change or specific variant in that gene can be evaluated. Variant classification is the process to understand the impact that a specific variant has on health and disease. In 2015, the American College of Medical Genetics and Genomics and the Association for Molecular Pathology published guidelines on how to classify what we would call sequence variants, these specific changes in the gene. This publication describes the pieces of evidence that we can put together to reach what we call pathogenic, which would be associated, likely pathogenic would be associated with a disease, or what pieces of evidence do we need to put together to reach benign, which would not be associated with causing a health condition. And then when might we be left in this kind of uncertain, this middle category, this uncertain significance, where we don't yet have enough information to kind of put that variant in a more definitive classification. We did receive some questions, and so I just want to address this about when updated variant classification guidelines may be coming out, and everyone is working on it, and it is still in progress. So obviously when they are available, there will be lots of announcements. To kind of put this in more of a visual display, this is one of the tables from the 2015 guidelines. And this table provides guidance on how to use different pieces of evidence when determining the classification of that variant. What pieces of information would move it to being very strongly associated with pathogenic, or what levels of evidence may make us feel very strong in that it's not associated, it's benign. So what pieces of evidence are being evaluated? So here I have highlighted some questions that need to be reviewed during the classification, during the variant classification process. We will discuss each of them in a little bit more detail, but this just kind of is a brief overview of what research is being done by these curators, by the clinicians, by the individuals trying to classify the variant. First, we want to know, has anyone studied this variant in a research or a laboratory setting? Experimental studies focus on understanding the effects of a variant. For example, scientists may examine the symptoms of mice with a particular variant, or they may want to understand the variant's impact on cell growth in a laboratory setting. In addition, we want to understand the impact that change has on the protein being created. Again, I'm going to reference our YouTube playlist that have many videos on understanding the different types of variants. But here is a quick example. We have a variant in a gene, and we want to kind of understand what is the predicted impact this variant could have on the protein. So is the variant that I was identified what we call synonymous or a silent variant? And these are changes in the DNA sequence, but that does not lead to a change in the amino acid, which it's unlikely to cause a change in the protein. So in this example, the sequence nucleotide C was changed to a T, but both code, the TCC and the TCT, code for the same amino acid for serine. Since they code for the same amino acid, it doesn't change the protein, and that makes it kind of unlikely to impact the protein function. So a lot of times, if we see a silent or a synonymous variant, we are leaning more towards this is not really strong evidence for this variant being pathogenic. It leads us to a little bit more of a, maybe this change is benign, maybe this change is unlikely to impact health. On the other side was the variant we'd see create what we would call a nonsense variant. A nonsense variant is expected to shorten the protein. So here again, it's the same situation where there's a single letter that has been changed. The G was changed to a T, and that did result in what we would call a stop codon. It told the body to don't read anymore. We have reached the end of this information to create a protein, and therefore this protein is now shortened in size. It's smaller than expected. And that may then lead to a change in how the protein works. Does it get to the right place? Do we have the right information? So understanding the type of variant that we see and its impact on the protein gives a little bit more of, okay, if it's a nonsense variant, it is in changing the protein size. This may impact the function. This may then impact the health of the individual. The next batch of questions kind of goes along, what do we know in sort of additional published literature and some of the more publicly available databases? So have other individuals with the same variant and similar symptoms been reported in the literature? Is there enough case examples? Meaning, have there been enough individuals reported that have a change in the gene and that they all have similar symptoms to maybe classify this variant as disease-causing, as likely pathogenic or pathogenic? Or has this same variant been seen in many individuals with no health conditions? So maybe leading us to a classification of benign that maybe is just more common in the general population. So what do we mean by population data? So this is a tricky question, but basically a cutoff that we commonly use is around 1%. if a variant has been seen in 1% or more of the general population, it is less likely that variant alone is causative of a health condition. If one specific variant is seen in more than 1% of the population, we would expect to see a large number of individuals with that specific health condition. Oh, I didn't mean to go back. For example, let's say we have an individual who has a history of epilepsy, intellectual delays, and was identified to have a heart defect at birth. This individual went through genetic testing, a variant was identified, and then we're like, okay, we found this change. Let's learn a little bit more about this change. When we compared this specific variant to some of the databases that have reported variants in individuals that don't have any health conditions, so don't have a history of seizures, that do not have a history of intellectual delays or any history of birth defects, we said that this variant has been reported in about 1,000 of those healthy individuals with no reported health conditions. Based on that information alone, we're going to kind of say it is unlikely that this specific change is impacting the individual's health. Again, if we had seen this change in 1,000 other people, we would expect that those people would probably have some of those same health conditions. They have epilepsy, intellectual delays, birth defects. And lastly, we need to ask questions that are really related to that individual who had testing to understand, is this change related to their reported health history? For an example, we want to know what type of testing was completed on this individual. Did this individual have testing that was robust, looking at multiple changes in multiple genes? Was this test really specific to just one gene? Were there any other variants reported in this individual, or was this the only change that was found? What's going on in this individual's family history? Does anyone else in the family have any symptoms? If no, does anyone else in the family have this change? Or if individuals in the family do have symptoms, do they also have this change? Do we see that this variant is, kind of aligning with the information we have from the rest of the family? So one of the key things that we look into is inheritance of that variant. And so one of the pieces of information that can be valuable is understanding if a variant is new or unique, or what we call de novo to that individual. So in this family history, we have the biological parents. They did not report any personal history of a health condition. When we did their genetic testing, no variants were identified. When we did test the individual who had a health history, say, of intellectual disability, of seizures, of maybe a strong history of muscle weakness, we identified a variant that was new or unique, considered de novo in that individual, a change just seen in that individual, not seen in any of the biological parents. Though that piece of information makes us a little bit more likely that this variant could be disease causing. So again, we have that ClinGen does have expert panels that work to provide additional guidance on how to classify variants in a specific gene. So these expert panels we call VSIPs. And so what they do is they really focus on providing additional information that can be helpful to laboratories, researchers, and clinicians when they're trying to review a gene. And then these groups also publish their own classification. So to put this into context a bit more, again, this is the same chart that I showed you before. I have highlighted one piece of information that can be included, and that's understanding of the patient's phenotype. And so these guidelines that have been created are kind of general. They are made to be broadly applied to more to a lot of the genetics, to a lot of genes. So what our variant curation panel experts will do is they will take these guidelines and they will flush it out. They will provide more detail, more context on what pieces of information can go into it. So for an example, if you are trying to understand variants in a gene that's associated with a metabolic condition or maybe a blood disorder, is there any additional blood work that can be done to kind of understand that impact of that protein? So for instance, if we're looking at genes that result in a protein that is what we would call an enzyme, do we see a change in the enzyme levels of that individual? If we see a change in the enzyme levels of that individual that has a specific genetic change, we can say, oh, we are thinking that this change, this variant, is actually impacting the gene function, the protein function. We're not seeing the enzyme levels at what we would call the typical level. We see that there are less enzyme levels. The body is not producing as much of that protein. Therefore, we think that this variant is more likely to be disease-causing based on these features. So the VSEPs really take the guidelines and they say, let's make it specific to the gene that we are studying. What aspects of our gene are unique? What aspects of our condition are unique? What additional information can we gather to help with the classification? Similar to the DSEPs, VSEPs are curating genes across to many disease spaces, as I just alluded to. The first step that the VSEP does is provide those additional specifications when evaluating variants in a gene. They test the specifications in a pilot program. Once they are happy with those specifications, they are published. We make those available so that other labs, other researchers, other clinicians who are trying to classify variants in this gene, in this disease space, have those additional resources, that additional guideline, that additional framework to help with their evaluation. After the framework has been established, the VSEPs then begin their own classification process of variants. They really try to focus on a lot more of the tricky variants, so maybe variants that have a conflicting classification between laboratories. So maybe one lab classified that variant as benign and another laboratory classified that variant as uncertain. Or they may see that multiple labs have classified this variant as uncertain, just kind of saying, oh boy, we really need to understand this change better. We really need to look at it and pull all of our resources together. Again, anytime a VSEP does make a classification on that variant, that information is made publicly available on both the ClinGen website and then into ClinVar, which is a public database of variants. So what does this all mean and how can you all help? So why can data sharing help with the variant classification process? So to better understand genes and variants and their relationship to health, it's important to pull as many resources together as possible. Individuals engaging in data sharing can be extremely helpful in providing additional case level data that can be used by these expert panels when gathering evidence for either their gene disease validity classification or the variant classification. As a quick reminder, participants in ClinGen's patient registry, GenomeConnect, consent to de-identified data sharing and recontact over time. They upload a copy of their genetic test results and provide their health history through patient surveys. Once genomic and health information are collected from our participants in the registry, this de-identified information is shared with ClinVar, which I've mentioned a few times. ClinVar is a publicly available database that is often used by laboratories, researchers, and clinicians during the variant curation process. Here is a screenshot of kind of of ClinVar, and all the way at the bottom, you can see a GenomeConnect submission. And so GenomeConnect does not classify variants ourselves. We report the classification that was on the laboratory. So how was the report you gave to us? What classification was provided by the laboratory that did the testing? What we do is we provide some additional contact information. So again, in this screenshot, we can see that there are 3 labs that have submitted this information, and we can see kind of our GenomeConnect submission as well. If I expand the GenomeConnect submission, you can see that we do provide some additional clinical information that can be helpful. We can provide information on kind of when was, at what age was the individual tested? Does that individual have any clinical features? And sometimes we can provide a little bit of that inheritance information. So I call this, on this page, is called the allele origin, but sometimes that inheritance information, as we said, do we know if this variant is de novo, can help with the classification process. So data sharing can provide additional details and information to kind of help answer some of these questions that are asked during the variant classification process. So first off, the more individuals that undergo genetic testing, the more information available to determine if this variant is frequently or infrequently seen in the general population, right? That generating that population data set is really important. To generate that population data set, we need people to have genetic testing. We need the variants to be identified. We need to know how common these variants or how uncommon these variants are. And then second, we kind of say, are there individuals that have similar variants in the same gene? So for an example, if the protein structure has been changed, right? If we see that other people that have a nonsense variant, that change a code or a stop codon in that protein, if that specific change has been already defined as being associated with a disease, then if I have another patient that also has a stop codon or a nonsense variant in this very similar region of the gene, that makes me suspicious that it would kind of have a similar impact. And so kind of putting that information together, if I'm looking at different areas of the gene, do I see that maybe a lot of people that have changes in this gene, have they also been shown to exhibit symptoms? So maybe this area of the gene is really important, and we really do need to understand any changes in that area. So other things that is really important that our participants really provide us is, has the variant been seen in anyone with symptoms? Was it inherited? What other variants were found through testing? So often laboratories submit to ClinVar in aggregate, which means they kind of submit the information kind of as a bundle. If they have seen this variant in a number of individuals, they are going to just submit it and say, we've seen this variant, this is how we classify it. They're not really going to provide all of that specific health information, the inheritance information of that variant. So that is what we would call case level data. They are submitting, we've seen this variant, this is our classification, and this is the date that we have this classification. Individuals engaging in data sharing can really help provide additional context, additional health history, all of those kind of questions that we just went through, and that may be helpful to the kind of variant curators as they are looking to understand this impact of this variant on health. So all this to say is patient data sharing can provide access to what we would call unpublished cases that can be used to inform gene-disease relationships, and they can also provide that case-level data details that can be really useful to inform variant classification. So now the next process is variant classifications. Are they stable always? Is there a possibility that they can change? So as you can imagine, as more information becomes available, our understanding of genetic changes can shift over time. We do hope to reclassify kind of uncertain changes as a more definitive category as benign or pathogenic. Our understanding of benign and pathogenic variants also might change over time as more information becomes available. One study of a cancer genetic laboratory found that in a 10-year period, 6.4% of the variants they found were reclassified, leading to almost 60,000 reports being updated. So we know that clinical laboratories and healthcare providers, there is some duty to reassess genomic variants and recontact patients, but who is actually responsible? How does this work? How will this workflow actually go about is really kind of an ongoing discussion. There's no clear legal duty to recontact that has been established. And but mostly we have decided, or there is a consensus that recontact is beneficial. But again, really, there's a lot of barriers. So laboratories and healthcare providers, there's limited resources. Maybe a patient moved, maybe they're no longer being seen by that provider. So kind of puts a lot of limitations on this recontact process. And so to kind of address this, we have made an option available through our registry that we can help facilitate genetic report updates. If through our data sharing process, we do learn of any possible updates to a genetic report, families can consent to receive this information. We often inform our participants via email. We say that there may be an update available, and we do really encourage them to contact their healthcare provider and request an updated report. There's a few things that we do like to highlight. We will not identify all variants, and we still do encourage participants to follow up with their provider, kind of ask for any additional information about their variants, kind of more routinely every year, every 2 years, etc. To date, GenomeConnect has shared over 430 report updates with our participants. And as you can see from this chart above, the classification changes have been seen with all categories. Some variants have maybe been classified from an uncertain result to a pathogenic, or maybe to a likely benign. We've had some variants that went from pathogenic to uncertain, and some variants that went from benign to likely benign. So kind of all we have seen changes with all different variants. If you do have questions about your specific test results, or you would like to see if there's an updated information regarding your variant, we do recommend contacting the doctor's office that ordered your test, or reach out to kind of a genetics provider. Your healthcare provider may be able to refer you to a local genetics provider. If you are unable to, if you don't remember your healthcare provider, or you want to talk to a local genetics provider, the National Society of Genetic Counselors does have a webpage or a search feature that can help individuals find a local genetic counselor that maybe they can reach out to, see if they can schedule an appointment to go over their report in more detail. So I am talking to you today, but ClinGen and GenomeConnect is a massive team. You've met 2 of our team members today. And so this is just a huge thank you to everybody that does all of this amazing work. And we really appreciate everyone attending. But we also like to thank all of ClinGen has a number of volunteers that help with the classification process. And so we are extremely thankful to all of the work of everyone that goes into it. So now I will take a look at some of the questions that we may have.
Thanks for that, Caitlin. We do have a couple of questions in the Q&A, so if I could tee a couple up for you, does that sound like an okay place to start?
Sounds great.
Great. So one attendee asks, can we go on ClinGen ourselves to find out whether our VUS is still a VUS? And if so, how to access it? So I think a couple of things there we can say more about.
Yeah, so absolutely. The ClinGen website does make everything publicly available. If you go to ClinGen, the clinicalgenomeresource.org, there is a big search bar in the middle if you want to search your gene. What I will direct you to a webinar that we did last year on how to track two things. There are two things you can do. You can track your gene to see if there's been any changes in that gene disease relationship classification. And we have a webinar on how to track your specific variant in ClinVar. So it goes through how to search ClinVar, that public database, to find your variant and how to make any and to get alerts if there's been any new classifications when they come about on that variant. So those are two webinars that we did in the fall of or autumn of 2024. And so I will direct you to our playlist to kind of learn a little bit more about those. But yes, absolutely, that information is available, and it is, we made features for people to track it so they can stay updated. Great question.
Lizzie just put that ClinVar webinar in the chat if you want to take a look. That will allow you to track your variant in ClinVar and see what are laboratories others submitting as the classification for this variant. It is a submitter-driven database, as Caitlin mentioned. It relies on people sharing data. So we do also always recommend that circle back with your provider for updates too. But this is a great way to remain up to date as well. Similar to that question, Caitlin, we had someone ask, how do we know if their variant was submitted by GenomeConnect? So could you say a little bit there as well?
Yeah, that is a great question. So on um, on that variant page, for the individual specific variant, if GenomeConnect has submitted, it will be down at the bottom and it will say it usually says GenomeConnect that it was submitted. You can always reach out to us directly. Our email address is on the screen, info@genomeconnect.org. With GenomeConnect, we do also submit an aggregate. So if you signed up today, your variant will probably not be submitted to ClinVar right away. It does take us a little bit of time to curate the reports, put all that information together, and then submit to ClinVar. We are also working on a better notification process for our participants, which we hope to implement soon, where we will send out an email saying that the information has been shared.
Thanks for that, Caitlin. We also have a really great question around AI and variant classification, and really thinking about how do we think AI is it going to be useful for variant classification? How is it going to be useful? Maybe what are some of the caveats, things like that. And I'm happy to give my perspectives as well, but I'm sure you have some, Caitlin.
I think it is going to be useful. I think part of some of the things that maybe it can help reduce some of the more manual checks, right? If we use AI to check the population data sets to say this how common is that variant and just kind of provide that information quickly. And so you can check off some of that information. We have been trying to work with AI to kind of pull in the publications that may be really useful when classifying it. So, using AI to find the papers that do have those case level data available, that maybe do have the experimental studies that really do kind of help gear us to the questions that we want to answer. I also think that maybe AI can be used to, as we said, to aggregate the information, pull in data from different resources a little bit more clearly. I do not think that it's going to replace our expert panels because our expert panels are really diving deep into those classifications that are a bit tricky and really do need the experience and teamwork and kind of everyone to put their heads together. But Joel's happy to hear your input as well.
I agree wholeheartedly, Caitlin. I think it's going to be really useful in pulling the information together, and then that extra layer of review by experts can be really helpful. So, completely agree. I think one area where AI in some ways has been involved in variant classification are what we call in silico models. And so it looks at how would it impact this protein? Do we think it's going to be damaging to how the protein works in the body? And so they have been a part of variant classification for many, many years, but it is just one piece of what goes into variant classification. We don't classify something as pathogenic based on those in silico models alone, but it is part of, as Caitlin mentioned, the pieces that we put together. There's also a question about what happens if a pathogenic variant turns into VUS or likely benign. And I think this person's asking this question of some genes, if an individual has a pathogenic variant, we recommend preventative surgeries. And so they're kind of wondering about, oh goodness, what if we do those things? I think I'd like to say that those downgrades from pathogenic and likely pathogenic down are quite rare. And that's because of how we approach variant classification and all of the pieces that Caitlin mentioned that go into classifying a variant. If we're going to call it pathogenic, likely pathogenic, we want to be quite certain that we think this affects how the gene works in the body. Now, I said quite rare. That doesn't mean that they never happen, but it means that for most variants that are classified as pathogenic, likely pathogenic, they remain in that category. Downgrades, if they do happen, those are things to work with your healthcare provider, genetic counselor around kind of what are the next steps, what's appropriate based on your results, but also based on other things like your personal history, your family history that can go into some of those discussions. So downgrades happen exceptionally rare from the pathogenic, likely pathogenic down. Caitlin, I don't know if you have anything else to add there too.
No, again, I think as a community, we are overall take a very conservative approach. We need lots of evidence before we feel comfortable classifying it as pathogenic, likely pathogenic. So again, very rare, usually very robust amount of data before that we provide that classification. And robust amount of data upheld over time, which is another piece of the puzzle, which sometimes can be a little bit both really great in these situations, but also can understand a little bit of frustration. And when you're looking to see if your variant's going to be reclassified, we really need to continue to gather this data over time. We need to study individuals over the course of their life as well. So it's really great. And also I can understand it being a little point of frustration at the same time.
Caitlin, I think we have one last question. This person clearly took a look at ClinVar, saw our submission, and asked some questions about the classification. So they ask, my variant no longer says unknown significance, but says not provided in the classification. Could you say a little bit more about how we submit to ClinVar and why that might be the case, and maybe where else to look for the classification as we saw it on the report?
Absolutely. So when GenomeConnect submits to ClinVar, we use a type of submission called phenotyping only. What that means is GenomeConnect is not providing its own classification. We will only report the classification that has been provided on the report. So how does this impact how it looks on ClinVar? It says that GenomeConnect itself did not provide a classification, which is true. We don't want our information to submit to that overall variant classification aggregate status. We don't want it to conflict with what the labs have been reported or the expert panels. The information that is gathered on the report from our participants is in that more category. So if you go down to that ClinVar submission and you click for more information, this box will kind of populate and it will kind of have additional layers of information such as the lab and the lab name, where was it tested, the report date, and the classification that was on the lab. But if you do have a specific question, feel free to email us at info at genomeconnect.org, and we can pull up the submission and highlight those pieces of information that you're looking for.
I think that does it for our questions, Caitlin. So I'll turn it to you maybe to close us out.
Yeah, so again, thank you all for attending. Thank you all for your questions during and before the webinar. We really do hope that it was helpful. As always, Lizzie had just put in our email address if you have additional questions. And we thank you all for your time today, and have a great rest of your day.
Thank you.